for the invitation to speak. I'm actually kind of surprised that we've gotten this far without a PRS talk today. I mean, there's been a lot of interest in polygenic risk scores, or renewed interest ever since the Kathy recent paper was published about a year ago, where they found that polygenic risk scores, and really published it for the first time, can be highly predictive of a complex phenotype. And they looked at a number of phenotypes, including coronary artery disease, that had in their hands the best score, and was shown to equate to monogenic equivalent risk. And that could then, at those levels, be used for clinical care. And so the discussion sort of turned fairly quickly from, how and when should this be applied in the clinic? But that sort of ignores one of the main sort of challenges with this, is that right now they're sort of adequate to be used in the clinic for a certain population, and that's that these scores are not very portable between populations. And so the aims that we'd sort of proposed for this study would be to delineate the heritability and generate polygenic risk scores across a range of phenotypes in different populations. So African Americans are a big constituent of our biobank, and that would be one population that we'd be interested in looking at. But in the context of this consortium, there are many other populations that we could also examine. We'd also be interested in looking at the effects of age on the prediction accuracy of polygenic risk scores. We are a pediatric biobank, and there are a number of phenotypes where the heritability varies with age. And finally, we propose to look at polygenic risk scores as biomarkers in a targeted therapy, and I'll talk about that in a minute. So the phenotypes that we've selected, the cardiovascular ones are an obvious choice. They've been shown to be very predictive and of potential clinical use. So if we generated these and validated them for other populations besides European Americans, besides European populations such as African Americans, they could be of immediate use. We've also selected a few others, such as Alzheimer's disease and neuropsychiatric phenotypes, primarily as to develop them as biomarkers for drug discovery. Taking Alzheimer's disease, for example, from 1998 through 2017, there were 150 trials. Only 4 of them passed, with 146 failures. The most recent of which, Biogen's beta amyloid monoclonal antibody, was reported last month. These 146, it may well be that some of these drugs are actually good, some of these mechanisms are good, but they're just looking at the wrong stage of disease. And so by having a polygenic risk score, you can estimate an individual's risk and perhaps target the therapy at a more opportune time. So what is a polygenic risk score? Well, essentially it's a fairly simple idea. It's in a complex disease, you have many genetic variants that are conferring risk. And this is a way of capturing that variation and distilling it into a score. You can use that score to then stratify patients according to their risk of developing the disease. And really what's driven this sort of the success recently is the development of some new statistical models. But the key really is the large GWAS sizes that we are achieving now. And these allow us to accurately estimate the risk that the variants are conferring. So this is a typical workflow. You start off with a training set. This is a pointer. Anyway, you start off at the top with a training set. Typically that's the results of a GWAS. Again, this has to be a fairly large GWAS at this stage to accurately get your variance. You then link that in with a linguistic equilibrium panel that's based off of the population that you're studying. So most of these are actually European ancestry panels. You then generate a number of different scores based on different parameters and different cutoffs, using different algorithms, if you like. And then the second stage is to validate those in a separate data set. And so the validation data also has to be a fairly large data set. And then you select the one that works the best, and you can then go on and test it. And so it's clear that polygenic risk scores and the success of those is highly correlated with sample size. And if we take a quick look at how that shakes out, how many samples in the GWAS catalog, for example, or how many studies have been done on the different populations, we can see that out of 3,900, 2,900 have been done in Europeans, which is about 75%. And then we have 27% in Asians, that number is growing, and 17% in Africans, African Americans, with the smallest number currently in Hispanics and Latinos. So that's just looking a rough look at the GWAS catalog by searching titles. Martin et al. Recently, in their recent publication just this month, they took a slightly different approach. They looked at the actual, each participant and what their descent was, and they come up with a fairly similar number. So they were looking at 79% of European descent. And a couple of years back, Pope Joy estimated 81% of all samples were of European descent. So there's clearly a high enrichment of European samples in the GWAS catalog, and that also means that samples that have been genotyped. So if you're developing a PRS and you need large sample size for success, it's clear that you're going to go to European populations to develop your models. So what that means is that these models are then locked into the European populations, and for numerous reasons, including essentially differences in allele. So there are in linguistic equilibrium. That then means that you're not going to be very predictive in other populations. So if you take a quick example, one of the best known genes in IBD is NOT2, and NOT2 was the first IBD gene to be discovered. There are 3 missense variants in there that confer large risk. And they're simply not present in East Asians. So if you go and look at your East Asian populations, that risk allele frequency is essentially 0. And so that's an extreme example, but you can then go into the NHGRI catalog again, the GWAS catalog, and identify all variants that have been associated in GWASs and in Europeans, and then check what those allele frequencies are in Africans. And when you do that, you can see that there's a systematic difference in allele frequencies. So there's basically a 12% increase in allele frequencies of the ancestral risk in African populations, and there's a concomitant decrease of 7% in the derived allele frequencies. Linguistic equilibrium can also result in population-specific differences in association. There's a couple of very well-known examples in IBD again, the TNF-SF15 locus, for example. There's increased variance in non-Europeans, and at ATG16L1, there's increased variance in Europeans. So the combined effect of all of these different, the LD, the sample sizes, etc., it basically means that we have polygenic risk scores that are predictive, but just in Europeans. So we took the risk score that was developed by Kathy Reeson for the IBD. They generated that in 12,800 cases and used 21,000 controls in 7 million SNPs. And they got an AUC of 0.63. We took that exact same PRS and applied it to our population of African American patients. Smaller numbers, we had 1,600 with Crohn's disease and 583 with UC, but still pretty good, and got an AUC that's pretty close to just being a random guess. So you can see there that that's just one example. There have been a few others reported in the literature. So the top table, we're looking at a BMI, educational attainment, height, and schizophrenia. The third column lists basically the R-squared in the European population that the tool was developed in. And the second column lists the equivalent in African Americans. And you can see that pretty much for all of them, even though there's some variation between studies, the trends are pretty much the same, that you have pretty much a 10-fold decrease in the R-squareds between the two populations. And this is not just restricted to African ancestry. Again, in Mark Daly's paper from this month, they looked at 17 anthropomorphic traits. These are basically blood cell traits from the UK Biobank. And they looked at the small UK Biobank African population and East Asians from Biobank Japan. And what they found was a systematic decrease in the predictive power of the tools in non-European populations. So there was a twofold decrease in East Asians, and that rises to a 4.5-fold decrease in African populations. So the age effects, there aren't really any numbers around it like there are for the ancestry. But what we do know is that heritability of some clinically relevant traits, including adiposity, BMI, lung function, and systolic blood pressure, varies with age. Particularly, the heritability declines with age, mostly due to the accumulation of environmental perturbations. And so pediatrics suffer from the same sort of issue as non-European populations, in that most of the samples that have been genotyped are in adults, and so it's an ascertainment issue. So there is concern that we're going to be using PRSs that are trained in adult populations and underestimating the effects in our pediatric samples. So this is something that we're interested in looking at. So those are the concerns. The way we are proposing to deal with this is essentially use a couple of different polygenic risk score models and then apply those in different populations. So relatively simple, really. So the study designs on the far left-hand side would be your traditional sort of model. You apply it in a European ancestry, validated in the European ancestry population. And that'll give you, due to the availability of large GWASs, potentially the most predictive scores, which will lay a good baseline for heritability analyses, but also in tuning the R-squared values. And it'll also be a good approach to assess the age effects. In the second model, we're looking at non-European ancestry cases. So this would be, for example, African Americans as a training set, validated in the same African American population, and then apply it across all. The reason that you want to apply it across all populations is, A, to get an idea of how the heritability and these scores are performing between different populations, but also with the hope that if you perhaps with the increased variance in African populations, that they may actually perform better in Europeans and East Asians. However, we do have the issue here that current sample sizes tend to be restrained. So the third possibility is basically to merge these two in a multi-ethnic training set. What we can do is create a very simple linear combination of two PRS scores that were carried out independently of each other with a weight. And that was an example of that was published in 2017 by Alzh Price's group. And when they did that, they found a pretty significant increase in the efficiency of the score, 30% in African Americans and 70% in East Asians and Latinos. So the actual leveraging of the consortium is in ascertainment of samples. This should not be a commutation of samples. That's a typo. So essentially, if we can accrue some samples for the phenotypes of interest, we have significant experience with a combination of genotypes and also of imputation through some studies that we've been involved in and actually sort of led in the eMERGE consortium. So in the eMERGE consortium, that's it's a study of several different sites, all with EMRs and with genotyping data. We've just gone through in this third phase an exercise where we've imputed almost 100,000 samples across the entire network. So that's 7 different sites that were genotyped on 77 different arrays, and we've managed to combine those into a single VCF file that can be then computed against. So we have that experience. We also have a significant number of minority samples already. So we're part of the TopMed program that's funded by the NHLBI, and as part of that, we have 7,000 samples that have been funded through CAG, but the entire consortium includes a significantly larger set. There's 45,000 African Americans now and 23,000 Hispanics, Latinos, and just under 14,000 Asians with whole genome sequencing data. Also, the CAG biobank includes 45,000 African Americans with bank DNA and mostly genotype samples too. So the other piece is the phenotyping algorithms. So again, this is leveraging work that's been done through eMERGE, whereby because there are these multiple sites, go through these exercises where if a site proposes a phenotype of study, they can build out an algorithm. Sort of that flow chart depicts a sort of complicated one, but the idea being that using a number of ICD-9 codes or 10 codes or diagnosis labels and medications, etc., data derived from the EMR, you can create a structured algorithm that then defines your sets of cases and controls. These can then be validated at a different site, and once you have an algorithm that's been defined, you can validate it through chart review and work out what your sensitivity and specificity are, both for the cases and the controls. And all being well, you can then deploy that pretty easily to other sites to define phenotypes in a similar way. So getting into the template parts of the study, the initial goals that we have really are to survey sites for availability of phenotypes and genotypes for non-Europeans, basically. If we can accrue significant numbers of samples for our phenotypes of interest, that's great. We would want to then look at what's been published. We can use published data to create our training sets to create the polygenic risk scores. We will need to account for sample overlap, and we can then combine any new data to boost those numbers from within the consortium whilst maintaining sufficient numbers for a validation phase. We will, in the phenotyping, so we may have some need to translate terms and use maybe some different billing codes, and the final challenge would be to consider where the analysis is being done. Ideally, sites could either send us some summary stats and we could carry out the analysis, or there is the possibility that we could also assist with imputation of the data. So what we would need in terms of data is either summary stats or genetic data that is derived from genotyping arrays, exome sequencing, or whole genome sequencing. Phenotype data would be electronic health record derived measures, and in terms of policy issues, as long as there is broad consent to share summary stats, that should be sufficient. So the cross-cohort needs, really it's open to all cohorts. Anyone who has non-European ancestry samples that have genotyping and phenotyping would be welcome to join. I've listed a number that sort of looked like they would fit that bill, and there's a number of them in this study. In terms of timelines, so we could start by evaluating the age effects in year one, regardless of whether or not, because this isn't dependent on accruing samples from minorities. So we would start in year one with the evaluation of the age effects, and then start to build out the phenotype definitions for some of the larger phenotypes, Alzheimer's disease and the cardiac phenotypes. That typically takes about 6 to 7 months. So that gets us into year two, where we would start to carry out the analysis from the phenotypes in year one, and then complete the neurophenotypes. And then in year three, essentially, would be completing the polygenic risk scores across all the different phenotypes. In terms of resources, this would be a relatively small project. I think 300,000 or 400,000 to fund a couple of postdocs would be sufficient, including some effort for participating cohorts to facilitate data transfer. There may be some costs associated with data storage and data common fees. And that's it.
I think we'll allow for one very quick question. Yes, please.
Just a slight editorial point. I think in these discussions, when we use we use the phrase European ancestry, we actually do ourselves a disservice, because in many of the studies, we exclude a lot of people who self-identify as European. The very early part of the GWAS, when we'll select the part of the European population you can model with LD, correct?
Correct.
And I just think it's not helping us in other conversations, because people think that the European population is a sort of scientific concept, when in fact, what we're modeling is this kind of well-mixed part of Europe, and certainly not everybody who identifies as European, in other words, people who would sort of self-identify under that. And I don't know what the right word is to use, but it's the kind of subset. It's a subset of Europeans that we're using.
That's a good point, and the word it's tricky, because Caucasian doesn't mean much equally. White also includes Hispanic, so it's a difficult sort of word to get right. But yeah, I take your point, and I glossed over the corrections of the ancestry-specific corrections that we would be undertaking. But really, the concept being that, okay, you're genetically selecting your ancestries in the first place, either way, regardless of self-report. And even though you have the north-south gradients and you're not sort of dealing with a homogeneous population, it is still controllable through stats. And it's not creating the sort of real sort of problems that you're seeing with these dramatic shifts in allele frequency that you would see between Europeans and Africans. So essentially, it's a label, and that's.
No, I get the stats. I just think the label actually it's a communication outside of the genetics community. In fact, to be honest, it's still a communication problem inside of the genetics community, where we think self-identified Europeans are the same as our study populations, and that's not actually true. And I think it helps us in the conversations with other populations like Hispanic or African American, when you say, well, the group of Europeans that we're studying is not all Europeans, it's actually a subset that we're looking at.
That's true, and then you also have to consider the European ancestry cases in the US and North America, so yeah.
Thank you. I think we'll need to move on to the next one.